In this last video, we're going to take a look at this process and what's known as the central dogma of biology. And a dogma is a belief that you're so sure about that there's no doubt that can enter your mind into the process. And what the dogma is that basically the genes with your DNA are going to encode the messages that are going to tell the ribosome what amino acids to put together and make for your protein. So this process of transcription and translation. And that was great for about maybe 10 years before a guy named David Baltimore figured out there was actually a reason that the central dogma was not true, and that's because there are certain viruses out there, with HIV being our cardinal example now. These viruses, the retroviruses, can actually take the RNA and run it backwards to actually encode for genes or for the DNA that can get inserted into your chromosomes as that provirus, and then that DNA is going to be used to be transcribed to make your messages that will actually be read by the ribosomes to make the viral protein. So like with everything in biology, there's always exceptions to the rule. But if we take a look at this process, remember, with transcription, you've got a promoter built into your DNA, and one of those strands of DNA is going to actually be used as the coding strand for your message. The opposite strand is the template strand that is going to be there basically for this process of DNA replication to actually proceed. And so if we look at this central dogma and we go into this process of transcription, we've got our promoter where our RNA polymerase would bind, and we're going to use this top strand as our coding strand. So this is going to be read by the RNA polymerase, and we're going to make the complementary RNA strand to our top strand right there. And so if you look, this is the same as our complementary DNA strand there in our template, with the exception that where you have your thymines, those are replaced with your uracil bases right there. And so if we look at this process right here, the RNA polymerase would bind to the promoter and transcribe this region of the DNA until it ran into the terminator, which I couldn't fit because I had my 5' and 3' labels there. But if we look at this, our RNA is made from 5' to 3'. There's your 3' hydroxyl end where you could add new bases if you need to. And I separated the message out right here into the codons just because it makes it a little bit easier to read. But if you're a ribosome and you're reading this message, there isn't going to be a little pause between these bases right here to let you know which one. You're actually going to have to identify where you're going to be reading this. The other thing that we can see here is this message actually gets processed in eukaryotic cells as it is being sent out of the nucleus to the cytoplasm for the ribosomes to read, and I'll show you that at the end of this. So if we look at this process of translation, and we look at the first stage here, the initiation stage, your small subunit of the ribosome is actually going to bind to the message around the 5' end, and it's going to start scanning along until it runs into the first AUG right there. There might be some other signals built in, so you don't have this AUG, does not have to be the first part of the message. There can actually be a long part of the message over here as the ribosome scans along until it finds this start sequence right there. When this start sequence is identified, the tRNA that is complementary to the AUG right there is going to float over, and it's going to form a hydrogen bond between the bases there, and that's going to hold the anticodon on the tRNA to the codon in the message. And this first tRNA, if you remember, this is the tRNA that is carrying that amino acid methionine there. So now we've got small subunit, and we've got our tRNA carrying this amino acid, and along comes the large subunit of the ribosome, and it's going to assemble on the small subunit with that tRNA actually fitting into our P site. So at this stage, our protein is effectively one amino acid long right there. And now we have started this process of initiation, and the next tRNA is going to be able to enter through the A site, and it comes into our large subunit. And I've gotten rid of the small subunit because it's not necessary anymore for the analysis of the process. The small part of it is really to clamp on to the message and to help identify that first start codon right there. If we look at this second codon right here, GAA, we go over to our table and read GAA, and there we have our amino acid glue, and that tRNA is going to enter your ribosome through the A site. So we've got our protein in the P site, and we've got our next tRNA carrying the next amino acid in the A site. And now your ribosome is going to start moving down the message right there. And when it does it, it's going to actually pick up that amino acid right here, and it's going to transfer it over to our next amino acid that was in the A site. So in this process right here, the ribosome moves down, and your tRNAs are going to be held in place. The ribosome activity that's associated with the large subunit here is what's known as a peptidyltransferase. So if you think about it, it's transferring the amino acid and it's making a peptide bond between those amino acids. So as the ribosome moves down, think about the first tRNA that carried the methionine, that is going to exit out of that E site right there. It can float around and get recharged by that tRNA synthase that is specific for this particular tRNA. That tRNA that was in your A site is now shifted over into the P site, and it is carrying your protein right there. And because this is in the P site, that means that the A site is available. And a new tRNA can enter into the A site, and here we can look, AAC is our next codon, and we come over to our codon table, and there is AAC, and it lines up with our amino acid asparagine right there. So this little guy is carrying the next amino acid in there. The first tRNA has been kicked out from our E site. And then the ribosome is just going to keep doing this over and over and over. Your ribosome moves down the message. The peptidyltransferase picks up your peptide and moves it to the next amino acid. So there we have our asparagine right there. And you've got your tRNA that is holding on to your peptide in your P site, which leaves your A site available. And you've got this little guy over here that can now exit. So we go, ribosome moves down, tRNA exits, you've got a free and open A site, and the next tRNA can come in. CUG is our next tRNA, which corresponds to our amino acid leucine right there. And this is one of the interesting ones, because if you look at it, you can see there's actually multiple codons with tRNAs that all carry the same amino acid. And this is what's known as degeneracy. There are multiple codons that can code for the same amino acid. You can see there's isoleucine and there's phenylalanine. Serine has these 4 amino acids, or these 4 codons in the message that would all correspond to tRNAs with slightly different sequences, but they all carry the same amino acid, serine right there. So your ribosome is just going to keep chugging along here, adding amino acid after amino acid after amino acid to your growing protein chain right there. And it's going to do that over and over and over as it's traveling along your message. And then at the end of this process, that ribosome is going to run into what we call the stop codon. So here you can see UGA. There's also UAA and UAG as stop codons. And the thing is, there's no tRNA that actually has an anticodon that matches that stop codon. So instead, a protein that's called a release factor is going to enter into the A site of your ribosome right there. The peptidyltransferase is going to pick up our protein and try and move it over here, but there's no amino acid in the A site right there. There's just this release factor. And because it's picked this thing up, your protein is now freed from your tRNA. When it hits that release factor, that's going to cause the ribosome to let go of this long chain of amino acids, and that will release our protein. This is also going to cause the small subunit and the large subunits of the ribosomes to fall off the message, where they can float around until they bind to either the same message or another message, and they will start the process of initiation and elongation all over again. And so when we look at this process, again, think about this codon table tells you the amino acids that are in the code or in that message right there. And remember what I said, some of these eukaryotic messages are going to have some changes that are associated with them. And these eukaryotic messages are actually going to get processed after transcription takes place. And it's kind of interesting, on the 5' end, they will add what's known as a modified G-cap, and you can see it's got a 7-methylguanosine right there with this triphosphate. And this is actually going to be one of those signals, they think, for the ribosome to be able to bind to the 5' end of the message. So it knows, hey, this is the start of the message over here where I need to start scanning until I find the AUG. On the opposite side of the message over here is an enzyme called poly-A polymerase is going to add about 100 to 200 A residues over and over and over. So you make what's known as a poly-A tail. And they think that messages that aren't full length will not have the poly-A tail, and that means you don't want to read that message because maybe it's truncated, it's short over here. And these messages actually will form a little circle so the 5' end can interact with the 3' end. So if you don't have your poly-A tail, that message is not stable and it'll get broken down really, really easily, but also the ribosome is going to ignore the message. The last processing that can take place is what's known as last processing is known as RNA splicing. And this is a really interesting process because these messages actually have a whole bunch of extra stuff that's built into them. And we don't want those sequences to be leaving or exiting the nucleus. And they're known as introns because they're actually going to stay inside the nucleus. So if we look here, there's our promoter, and there is our terminator right there, and built into the message, there's one exon, a second, and a third one. So these exons are going to get connected together, and the introns are actually going to get removed from the message as it's processed. You get your cap, you get your tail, and then these exons are going to get spliced together, and now that final message is going to be transported out to the cytoplasm where it's going to be translated. And this is really kind of a weird thing that had people scratching their heads and what's going on here. But if you look at what can happen when you splice those exons together, it could be exon 1, 2, 3, it could be exon 1, 3, 2, 3, 3, or just 2. All of these come from the same gene, but if you think about it, each of these could have slightly different sequences or slightly different codons in there, which means one gene could actually code for multiple types of proteins. And this is one of those differences that we can find when we look at prokaryotic cells and compare them with eukaryotic cells. So think about the differences because of the structures of the cells and how we can see differences as a result of this stuff. What's going to happen with DNA and replication, transcription of your RNA, and also translation of those messages. When we look at our prokaryotic cells, all of this is going to be taking place in the cytoplasm. Your single circular supercoiled DNA is replicated in the cytoplasm. Your genes are going to get transcribed starting at the promoters there, and the messages that come off of there actually can have what's known as polycistronic messages. One message can actually have 3 or 4 different regions in it that are protein coding regions there. And so this polycistronic means many messages are built in there. And this translation can occur in the cytoplasm, and you can actually get ribosomes assembling on the message as it's being transcribed there. And so you get these polyribosomes. This is translation occurring along your message there. When we look at our eukaryotic cells, they don't have this process. They make what are known as monocistronic messages from that linear chromosome in the nucleus that is wrapped around your proteins to form those nucleosomes that I talked about in Chapter 10. There's 3 types of RNA polymerases in our eukaryotic cells, and they're huge. They've got 40 to 50 different proteins. These are polymerases make monocistronic messages that are processed, and splicing can give us multiple different types of proteins.